Good afternoon, friends. Today we are starting with biotechnology. There's one more booklet for space that you would get, but I'll do it in an extra class, not as part of these 12 classes that I have. In the next two classes, that is today and tomorrow, I want to finish biotechnology. Okay, now any doubts from the previous class? Yes, Kapish.
So we have read that Chandrayaan 1 was an orbiter. Chandrayaan 1 was an orbiter, but in the key differences we are reading that it contained an impactor module. And why will it contain an impactor module for Rna if it is an orbiter?
If it's an orbiter, no. What happens is after its life is over, it is supposed to work for a particular number of years. There is no point of keeping it in that orbit. So it is made to impact on the surface of the moon and get destroyed. That is the impactor. Yes. So Kapish asked a question that Chandrayaan 1 was an orbiter program. Then why did it have an impactor? The impactor's role was once its work was done, it will allow it to impact on the ground. So technically speaking, impactor would be nothing but something to slow down the speed of this thing, Chandrayaan. The moment it slows down, it will fall. Like if the speed is less than under root gm by r, what happens? It falls. Speed is equal to under root gm by r, it will be in a circular orbit. Speed is more than under root gm by r, it will be in an elliptical orbit. Simple. First day's fundamentals are relevant forever. Okay, so let us start with biotechnology. Okay. At the end of the booklet, you also have a few questions that we should be practicing for it. Now, what do we mean by biotechnology? First basic thing, like when you hear the term biotechnology, what do you understand? See, what is technology? What is technology? Anyone? What do you mean by technology?
Application of science.
Application of science to develop something which is useful for human beings. Right. Biotechnology is a similar thing where we use biological processes, we use biological organisms, we use biological systems to develop something that is useful for human beings. This is biotechnology. So if you look at the definition, it is the use of biological processes, organisms or systems to manufacture products intended to improve the quality of human life. Did you understand this? Now, a very simple example of biotechnology would be curd. We all eat curd. Now, in curd, how do we prepare curd? We add some some living organisms, some bacteria in the milk. So, we used biological organism to prepare something that is useful for humans. That is an example of biotechnology. A very simple, very basic example of biotechnology. Genetically modified crops are also example of biotechnology, but this is also biotechnology. Fine. Did you understand this? How do we define biotechnology? Now, biotechnology can be classified into two broad categories. One is classical, traditional, old biotechnology, and other is modern biotechnology. Now, how is this classification made? If when you are using organisms, let's say when you made curd, what happened? You took some living organisms, you took some bacteria and used these bacteria to make curd. This is what you did. Here you did not modify the bacteria in any way. You did not modify the bacterial properties in any way. This is traditional biotechnology, old biotechnology, classical biotechnology. Suppose you had made some changes in this bacteria to increase the growth of bacteria rather than it taking 12 hours for making curd. This increased growth could have allowed curd making to be possible within 1 hour. If we had modified the inherent property of the bacteria, then it would have become modern biotechnology. If we had done some gene modification, then it would have been an example of modern biotechnology. Are you able to understand what I'm trying to say here? Use any process, use any organism. If you are not modifying the living organism, you are just using them. It is classical biotechnology. Curd making is an example of classical biotechnology. Making of alcohol. Yeast is used to ferment alcohol, right? There also living organisms are used. That is an example of classical biotechnology. Or making of, let's say, penicillin. Now, how is penicillin made? There is this penicillium, a living organism, a mold, penicillium. Scientists know that it produces penicillin. So they developed a mechanism to grow this mold in large quantities, extract penicillin out of it and sell it. Here, did we modify the mold? No. The living organism was used as such to produce something that is useful to humans. So even development of penicillin is an example of classical biotechnology. Got it? Understood? Curd making, alcohol making, even penicillin making, making of cheese, making of bread. In all these processes, biological processes are used. In all these things, biological processes are used, and they are example of classical, traditional, or old biotechnology. Now, let's talk about modern biotechnology. In modern biotechnology, we modify the inherent characteristics of the living organisms that we are using. For example, we know about cotton. Cotton plant comes from cotton seed. If you change this plant by genetically modifying it, you create what is known as genetically modified cotton. This genetically modified cotton may have certain advantages. For example, it may not be impacted by some pest which was impacting this cotton. Here what we did, we changed the inherent property of the plant. Tomorrow, like when we will talk about application of biotechnology, tomorrow we'll talk about GM crops, how GM crops are exactly made. Here we change the inherent property. It becomes GM cotton. Now see, suppose you extract algae for producing biofuel. Algae can be a source of energy. You extract it, and using it you make biofuel, you make biodiesel. What will be what type of biotechnology would it be? Classical. We have not modified the algae. The inherent properties of algae were not changed. But suppose you genetically modify algae to increase its rate of growth, because we'll need more biomass to produce more biofuel. If you modify the inherent property of algae to increase the rate of growth, it would become what? It would become modern biotechnology. You're understanding the difference? So here in modern biotechnology, we manipulate the genome or innate capabilities of organisms for making them more suitable for our requirement. Now, another thing, tissue culture or cell culture are also used in this system. Now, let's come to, we are not spending a lot of time in traditional biotechnology, we'll come to modern biotechnology. Okay, the basis for modern biotechnology is this. We having ability to modify a living organisms. Like I give this example every year. Suppose my father wanted that my son should have had golden hairs. Suppose this is what he would have wanted. He could have done genetic modification of me, modified me. Some genes could have been modified. He could have taken some genes from golden langur. Golden langur is a species mammalian species which is found in Northeast India. The gene which is responsible for golden color of golden langur, he could have introduced it in me. This is possible because in all living organisms, the genetic material is DNA. Basically, let's start with humans. Okay, in humans, we know that we are made up of various organs, right? Our heart is an organ, our lung is an organ, our skin is an organ, right? Our eyes, everything. We humans are made up of organs. Those organs are made up of tissues. Those tissues are made up of cells. Cells are the fundamental unit of life. Let me draw this cell. Cells are the fundamental unit of life. That is, we are calling cells as fundamental unit of life because a cell can survive at its own. Whatever is required for life to happen, eating of food, excretion, multiplication, all these things a cell can do. And that's why a cell is considered fundamental unit of life. Inside cell, there are various organelles. There is one nucleus, and then there are various organelles like Golgi apparatus, endoplasmic reticulum, all those things. Don't worry about that. We will cover those two. We will cover biology, like in 2 days. In next 2 days, we are doing biotechnology. Then we will go to fundamentals of biotechnology, fundamentals of biology. There we will study it. But coming back to this, cell consists of nucleus, and in this nucleus is our genetic material. Why are we like this? Why am I like this? Why my skin color is this? Why my hair color is this? All these things are there in my nucleus. All this information is there in my nucleus. Based on this information, my cell is producing certain kind of proteins. My hair color is of this color. Like, is my hair color similar to Kashi's hair color? There will be some difference because this is what is stored in, this information is what is stored in your nucleus. In my nucleus, some other information is stored. Fine, this is understood. So how we are, what kind of voice I have, what is my height, what is my size, a lot of it will depend on the information which is stored inside the nucleus. So this is cell and this is nucleus. And in nucleus, where is this information stored? This information is stored on what is known as chromosomes. Chromosomes will be this X-shaped structure which is there in your nucleus. You must have heard about chromosomes so far. Some chromosomes come from our father, some chromosomes come from our mother. So we have characteristics of our father and mother. So cell, inside the cell is the brain of the cell, nucleus, where all the information is stored. In the nucleus, we have a lot of chromosomes, and these chromosomes have come from our parents, mother and father. And it is here that information is stored. On these chromosomes, information is stored in the form of DNA. On these chromosomes, we have, what the hell? Why did it become eraser suddenly? On these chromosomes, information is stored in the form of DNA. Like DNA is rolled over it. This is DNA. How does DNA actually look? DNA looks like this double helix structure. If you the DNA which is there here, if you open the DNA, it would look something like this. DNA would look something like this with DNA would look something like this. So again, See, a lot of you are not from science background. It's important that you understand the basis of biotechnology. Once you understand these things, no, it would be very easy for you to read any newspaper article, if you look at a question, you will be able to, even if we have not studied those questions, you will be able to interpret. So, in human beings, we have cell, we have nucleus, we have chromosomes which come from our parents. On those chromosomes are these DNA. These DNA, though I have made it like this, you can understand it in this manner. Let me open that DNA. Rather than this spiral wala structure, let me make it like this. And then this is the structure. Same structure I have twisted. That is the structure. It occurs like that, but for simplification purpose, let's detwist it. Let's open it. This is how our DNA is looking, right? On these DNAs are made up of what is known as nitrogen bases. जैसे adenine is a nitrogen base, thymine is a nitrogen base, AT, or cytosine, guanine, those are other nitrogen bases. AT, AT, CG, CG, AT, CG, CG. ये order अलग है. Like in every human being, this order would be slightly different. Did you understand so far everything? Cell, nucleus. Inside the nucleus are chromosomes. Some chromosomes come from our mother, some chromosomes come from our father. On those chromosomes, we have this DNA. These DNAs are made up of nitrogen base pair. Basically, DNA को हम बोलते हैं कि it is a polymer of nucleotide, and a nucleotide consists of one nitrogen base pair, one pentose sugar, यहाँ पे, it is sugar, and one phosphate group. This is nucleotide, and DNA is a polymer of nucleotide. Polymer means a chain, a lot of nucleotides together. DNA is polymer of nucleotide. We'll come to that. But for now, did you understand everything till here? Cell, chromosomes, cell, nucleus, chromosomes, DNA made up of these nitrogen bases. Now, what is a gene? Gene is the fundamental unit of heredity. That is, one gene stores information about one protein. Gene is the fundamental unit of heredity. It stores information about one protein. That is, let's say 8 of these base pairs, nitrogen base pairs हैं, 8 of these steps together are encoding information. 8 of these steps are encoding information for protein 1. This DNA is very long. It has millions of base pairs. Billion, it may have billions base pair, more than a billion base pair. But a few of these base pairs are having information related to one protein. Another set of base pairs are having information related to another protein. This group of base pair, which is having information related to one protein, this subsection will be called a gene. Let's say this is the part which is storing information related to one protein. This is one gene. This is the part which is storing information related to another protein. This is gene 2. And so on. So gene is the fundamental unit of heredity. Gene is storing the formula for the protein, which protein would be formed. So my hair color are like this because a particular protein is being formed in my body, and that particular protein is being formed in my body because a gene related to that protein is there in my body. My cell produces that protein for which encoding is there. Everything is clear till so far? You're understanding everything so far? Now, water. Basis of modern biotechnology. We were talking about what is the basis of modern biotechnology. Basis of modern biotechnology is this. All living organisms, listen carefully. All living organisms have DNA as their genetic material. That is, in all living organisms, what protein would be formed will be provided, stored in a structure like this, except some RNA viruses. For example, COVID is an RNA virus. In COVID, what protein that SARS virus is producing, that virus is producing, is stored in RNA. Except some RNA viruses, all living organisms store what protein they would be producing in what is known as DNA, which are made up of ATCG only. Human beings, a bacteria. Even the bacteria has its genetic information stored in it in the form of DNA. Even the bacteria has that. Think of any living organism. Shashi. Shashi is a living, what's his name? Kashi. Kashi is a living organism. Suppose Kashi touches a plant, any tree. How is genetic information of that tree stored? It is also stored in the form of DNA. A bacteria which Shashi is adding in the milk to convert that milk, Kashi is adding into the milk to convert that milk into curd, also has DNA, that is ATCG base pairs. Now, if that bacteria also has this kind of DNA, though its encoding would be different, it will be producing some other protein. We may be producing some other protein. I understand it like this. In computers, how is information stored? 0101 in digital form? In computers, in your mobile phone also, this is the way information is stored. In supercomputers also, this is the way information is stored. In the digital watch that you wear, this is the way 01 ke form mein. In the same way, in all living organisms, information is stored in the form of DNA. And that's why, suppose golden langur, how is information in golden langur stored? Like this only. Suppose we identify, okay, this gene 1 is responsible for the golden color of the hair. Human beings also have this kind of DNA. We can extract this gene, we can take this gene from here and insert it in my gene, my DNA. Now, because the structure is same, suppose human beings' DNA would have been something like this. Its structure would have been something like this. Would it have been possible to insert this gene here? No. It's possible to insert this gene in our gene because our structure is also same. Our genetic information is also stored in the form of DNA. It's something like you can take a brick out of one building and insert it in some other building because both the buildings have the same type of bricks. If this building was made up of big stones and you would have wanted to extract brick from one building and put it there, it would not have fit. But in here, it fits because all living organisms are using the same. If you consider these things to be the bricks, and this is the overall structure, all living organisms are having this system. And that's why what happens, if you want to take a gene from bacteria and insert it in you, it is possible. If you want to take a gene from bacteria and insert it in a plant, it is possible. BT cotton. In BT cotton, what we did, cotton plant mein, this is a soil bacteria, Bacillus thuringiensis. We took a gene out of this bacteria because this bacteria, that gene produced a poison which killed pest. So we took that gene and we inserted it in the plant only. Now that plant produces that poison which kills pest. Why is this possible? This was possible because in this bacteria also this is the structure. In this plant also this is the structure. You're getting my point?
So every living organism has genes, like ultimately
DNA on DNA genes, yes, except some RNA viruses. So you will not be able to take features of those RNA viruses this easily. Except some RNA viruses, all living organisms have this structure. All living organisms have DNA, and on DNA they have genes, and those genes are encoding for their protein.
So does evolution plays any changes in gene transformation?
Yes, evolution, we'll talk about that too. Okay, but did you understand this? Now if you are asked a question, can you transfer a gene from mango tree to orange tree? Can you transfer a gene from mango tree to orange tree? Yes, why not? Why not? From mango to orange, can we transfer a gene? Yes, in mango also this is the DNA structure, in orange also this is the DNA structure. Take out a part of their DNA, that is take out a gene and insert it in the mango one. Can we do it from human beings to plant? Yes, human beings have this kind of DNA, plants have this DNA. Take a part out, insert it somewhere. Because each one of us are having the same DNA structure, mixing and matching of DNAs is possible. So a question was asked by UPSC a few years back where they said gene transfer is possible among which of the following pairs? Plant to animal, animal to bacteria, humans to fox. Humans to fox वाला option नहीं था, but yes, it's possible. Humans to plants, plants to humans, yes, it's possible. Because everyone is using DNA.
So is there any example of
No, we haven't done it yet.
but it is possible?
Yes, bacteria to plants we have done. Like we are not creating mixture of human, वो ethical वाले aspect आ रहे हैं. We haven't done that, but yes, it's possible. Biotechnology is a fascinating field. You got it? Once you understand this fundamental, rest of this chapter would be very easy, very simple. So if you have not understood it, go home, try to go through this section again. Okay. So most living organisms have DNA as their genetic material. DNA is deoxyribonucleic acid, the DNA that we had created, and therefore mixing and matching of DNA among different living organisms is possible. Now, some basic understanding, this I explained you. Nucleus, inside the nucleus we have, a cell, inside the cell we have nucleus, in the nucleus we have what? Chromosomes. Some chromosomes came from our father, some from mother. On chromosomes we have DNA. They have opened that DNA wala part, and on DNA we have gene. This gene is encoding for a protein, and in almost all living organisms this is how protein is encoded. फाइन, ये बात समझ में आ गई? सबको अच्छे से समझ में आ गई? नाउ in humans, यहां देखिए, genes vary in size from a few hundred DNA bases to more than 2 million bases. That is, one gene which is producing a simple protein may be made up of few hundred base pairs. Bases का मतलब base pairs, ATCG, ATCG. It may be made up of few hundred base pairs, or it may contain millions of base pairs. A complex protein would require more complex information being put there, so it would require millions of base pairs. Fine, this is also understood? Okay. See, the Human Genome Project have estimated that humans have 20,000 to 25,000 genes. 20,000 to 25,000 genes does not mean 20,000 to 25,000 base pairs. base pairs we may have more in them. Like every gene is made up of hundreds of base pairs or even millions of base pairs. So human beings have around billions of base pairs. Okay, now what is gene mapping? What do you understand by gene mapping? Gene 1 is responsible for which protein? Mapping this is gene mapping. We said that gene is the fundamental unit of hereditary. One gene is encoding for a protein. So this identifying the genes and which protein is it encoding for is what is known as gene mapping. Fine, understood? Okay. Now यहाँ पे we'll spend some time in understanding DNA. Like at the end of this booklet, we have around 20 questions. We will solve all those 20 questions. Those 20 questions include some questions of past year, some other questions to practice. Therefore, carefully go through each and everything that we have studied. I gave you a brief idea of DNA and all. Here we'll look at DNA in greater detail. So nearly every cell in human beings have DNA. Every cell has and it has the same DNA. Please understand this. Mother's ovum, father's sperm. It brought chromosomes, fertilized egg. One nucleus, one fertilized egg. In that fertilized egg, in its nucleus, we have chromosomes of both father and mother. Now this fertilized egg has multiplied and all our cells have been formed from this egg. So almost all our cells contain the same DNA material. Some of it do not contain nucleus, that's different, but otherwise if it contains nucleus, so if you take one human being, one cell of mine, you can identify what is my DNA. Got it? Now, most of the DNA is located in nucleus. Most of the DNA is located in nucleus. But there are some DNAs which are located in what is called mitochondria. So we'll study about mitochondria. Day after tomorrow, when we'll move on to biology, we'll study about mitochondria also. You must be knowing about mitochondria. Mitochondria is considered the powerhouse of cell. Right? It also contains some DNA. But the mitochondrial DNA comes from mother. Mitochondrial DNA comes from mother and it has no role in hereditary. What your features would be will not be decided by this mitochondrial DNA. So it's not relevant here. We'll study about mitochondrial DNA in three parent babies and all in the health wala sector of science and technology, but here we are not going to deal with it. Now, the length of DNA is usually measured in the number of base pairs you have, and in human 3.3 into 10 raised to 9, that is 3.3 billion base pairs is something that we have. 10 raised to 9 is billion, 10 raised to 6 is million, 10 raised to 3 is 1000. In this way, don't be Sambit Patra. You should know what is meant by 5 trillion. So trillion would be 10 raised to 12. Right? So these many base pairs is something that we have. Now structure of polynucleotide chain. Structure, see, this is the same structure that I had simplified for you. Open. Sorry. This was the vertical part of the DNA and this was the horizontal part. Now you may think that it is complex. It's not complex. It's very simple. Let me simplify it for you. GC, base pairs, which was forming the horizontal part. These are the nitrogen bases. Pairing of the nitrogen bases have formed the horizontal part of DNA. Right? In the vertical part, base pair is attached to a sugar, pentose sugar. And this pentose sugar is attached to the phosphate group. This thing together this thing, one base pair, one sugar and phosphate group is what is known as nucleotide. And DNA is a polymer of these nucleotides, deoxyribonucleic acid. You'll see this is what it is made up of. Nitrogen base, sugar, phosphate group, nitrogen base, sugar, phosphate group, nitrogen base, sugar, phosphate group, nitrogen base, sugar, phosphate group. DNA is a polymer of these nucleotides, which is made up of nitrogen base, sugar, phosphate. Even on this side, nitrogen base, sugar, phosphate group. ऊपर कर देते हैं. Nitrogen base, sugar, phosphate group, nitrogen base, sugar, phosphate group, nitrogen base, sugar, phosphate group, nitrogen base, sugar, phosphate group. So DNA is a polymer of these nucleotides, a chain of these nucleotides. ये यहां पे कहीं लिखा होगा structure of polynucleotide chain. So nucleotide has three components: a nitrogen base, a pentose sugar. In this case, this pentose sugar is deoxyribose, and therefore DNA is called deoxyribonucleic acid, because sugar is deoxyribose, and a phosphate group. Okay, now these nitrogen bases which are here, these are of two types. One is purine, other is pyrimidine. Purine consists of adenine and guanine, A and G. Pyrimidine consists of cytosine, uracil and thymine. But in DNA, we only have 4 of these nitrogen bases. Which one? Adenine, guanine, cytosine and thymine. A will pair with T and C will pair with G. ATCG के base pairs दिख रहे हैं. In DNA, we do not have uracil. Uracil is found in RNA. In RNA, thymine is not there. In RNA, uracil is there. So we'll come to this thing. But these are the nitrogen bases which are there. So now, can you again in your head, for those of you who especially are not from science background, try to create a picture of how DNA is formed, or like what is DNA? Inside the cell is the nucleus. On nucleus is chromosome. On chromosomes we have this DNA, and this is how it is formed. Why it is being called deoxyribonucleic acid? Because the sugar is deoxyribose here. In RNA, sugar will change. In RNA, sugar will be ribose sugar, not deoxyribose sugar, and that's why RNA will be called ribonucleic acid. DNA is deoxyribonucleic acid, RNA will be called ribonucleic acid. We'll come to RNAs also. This is understood? Two types, purines, pyrimidines. Adenine will pair with thymine and cytosine will pair with guanine. One more thing that you can note, adenine and thymine, when they are pairing, there are 2 bonds. When CG is pairing, there are 3 bonds. जो pairing हो रही है, CG 3 bonds, AT 2 bonds, A and T वो. तो
what was CNG? Guanine and
cytosine and guanine. Yes. Okay, now let's go to this. Also I have explained to you, adenine forms 2 hydrogen bonds with thymine and guanine forms 3 hydrogen bonds with cytosine. Okay, now how is information stored? How is information stored in the sense, like gene is storing an information related to XYZ protein. How is that information stored? To understand this, understand this aspect. Let's say you have to store 0 in a binary system where you have 4-bit system. How will you store 0? 0, 0, 0, 0. This is how you will store 0. In a binary system, you have to store 1. 0, 0, 0, 1. You have to store 2. 0, 0, 1, 0. You have to store 3. 0, 0, 1, 1. You can store different numbers using 0, 1 only. This is encoding for 3, this is encoding for 2, this is encoding for 1. Suppose you wanted to store 16. What would you have done? In 4-bit, only 15 could be stored. So you would need one more bit, right? So 16 in binary system would have been stored. This is 16. Right? Different numbers you could store in binary like this. In the same way, different proteins could be stored with the help of AT-CG base pair. Assume AT to be 0, CG to be 1. For now, so for simplify everything, AT is 0, CG is 1. Now you have a series of 0, 1, 1, 0, 0, 0, 1, and a series of this is a gene. A set of this is gene. Got it? How information is stored? Information is stored in these base pairs using a set of these base pairs. So human DNA consists of these two types of base pairs and information is stored using this. Now some more things you can know is like human DNA consists of 3 billion bases. 3 billion bases. How many genes? Around 20 to 25,000 genes is what human beings consist of. The order or sequence of these bases determine the information available. If the order is 0, 0, 0, 0, information stored is 0. If the order is 1, 0, 0, 0, information stored is 8. The order of these base pairs are basically determining which protein is encoded in your DNA. Now, should we first do some questions before we go to next topics? Now, I want to understand how much of these things have you been able to understand. Let's go to the end of the booklet. We are having some past year questions as well as some questions which I have prepared for you. Okay. Let's go to question number 1. The production of which of the following is our examples of modern biotechnology? Curd, wine, penicillin. इनमें से किसका production is considered? None of this. Penicillin में क्या था? वो mold था. Did we modify that mold? No. That living organism was used just like that. If we are not modifying the properties of that living organisms, inherent properties, so it's not modern biotechnology. It is classical biotechnology. So answer is D, none of the above. Okay. Let's go to second question. Consider the following statements about DNA. DNA is a polymer of ribonucleotides. DNA is a polymer of deoxyribonucleotides. पहले आपको बहुत difficult लगता, but it's very simple. You have to understand it from fundamentals. One is wrong. In humans, DNA is only located in the nucleus of the cell. DNA. Is it only located in the nucleus? It's also located in the mitochondria. Though it is not playing a role in hereditary, but it's also located in mitochondria. 2 is also wrong. The order and sequence of base pairs on DNA determine the information available for building and maintaining an organism. Right? Order and sequence will determine the what gene do you have. So what is the answer here? 3 only? C. Right? You're able to answer it? No? See, what I want is after you get out of this classroom, for after you watch this video, you should feel like, yes, today we understand biotechnology. Today we understand its fundamentals. We are not completely blank. Let's go to the next topic. Consider the following statements about DNA profiling. As a DNA profiling is not something that we have studied yet. We'll come to that. Dark DNA bhi abhi nahi padha. 2 questions, but through these 2 questions, I hope you have understood some aspects. Let's go back to DNA profiling, DNA fingerprinting, DNA testing or DNA typing. What is DNA profiling? और DNA fingerprinting की बात करते हैं. What is fingerprinting? Do you know? Fingerprints are considered unique. Those are used to identify individuals. In the same way, when you look at me, when you look at Amit, we people are different, no? So though 99% of our DNA would be same. By the way, before we come here, if you will consider all human beings around you, no, 99% of our DNA are same. We are producing same proteins. Only less than 1% of DNA differences are there between various human being community, human being groups. But still, we keep fighting in the name of religion, in the name of caste, in the name of country, in the name of color. 99% of our DNA is same. We are producing same proteins. We have evolved in the same manner. If you'll understand this now, you'll understand like हम कितनी छोटी छोटी चीजों पे लड़ रहे हैं. Like we consider other person inferior in spite of the fact that his body is doing 99% of the, each of his cells are doing 99% of the same thing that we are doing. We tend to discriminate people against on the ground of color or on the ground of various features, in spite of the fact that 99% of their features are same as our features. Because eventually it's the cell, the nucleus, the genetic material which is there, it is same. So let's stop discriminating on the ground of caste, sex, religion and whatnot. Understand this aspect. Okay, now come to DNA fingerprinting. What is DNA fingerprinting? See, fingerprints you can identify a person, no. In the same way, if you collect DNA, you'll find that DNA of different people could be differentiated. My DNA, using my DNA, you can identify me. My DNA would be different from your DNA. Though 99% of it would be same, but still differences can be identified. So that is, DNA profiling is forensic technique used to identify individuals by characteristic of their DNA. Okay, now let's understand what dark DNA is. What is dark DNA? Dark DNA was something that was in news last year. So while studying DNA, we'll also briefly look at dark DNA. So what scientists found recently was this. They did gene mapping of a particular bird or rat and some other organisms. Gene mapping, like gene 1 is responsible for protein 1, gene 2 is responsible for protein 2, gene 3 is responsible for protein 3, and gene last is responsible for protein last. Scientists did this gene mapping of some living organism. Genes are encoding protein, right? When they did this gene mapping for some living organism, for some birds, for some rats and all, they found that they have not been able, genes for some proteins are not there. In the gene mapping technique which they used, when they went through the DNA and identified all the genes, they found that genes for some proteins are not there. Those genes are not there. But these proteins are very important for survival of those living beings. How are they surviving? They found that these proteins are present in them. That's why they are surviving. But their gene was not there. In the gene mapping technique, the gene was not found. So what scientists understood by this is, our current gene mapping techniques are not good enough. There are some more genes which are there, which we have not been able to identify. Based on this, these proteins are being formed, because these proteins are important for the survival of those living beings. And that's why these proteins are being formed. But we have not identified these genes yet. That is, our gene mapping technology is not good enough. So these genes or set of genes are called dark DNA. Why dark DNA? You know about dark matter? It is said that the universe में around 20 to 30% of the matter is dark matter, which our current way of perceiving things have not been able to identify. Basically, how did scientists come to know about dark matter? It was this. They did calculation. I'm talking about dark matter now. They did we know mass and all those things. Using that, they did calculation. They realized that these galaxies would not have held together with the mass that they have. There must be more mass, more matter, which we have not yet been able to identify. More gravity is required for them to be held together. There should be more mass for that purpose. So we haven't identified that mass, but there is this dark matter which exists. Our current technology is not able to identify this. In the same way, these proteins are being formed, so these genes must be there. But our current technology has not been able to identify or map these genes. These genes together are called dark DNA. Got it? Did you understand what is dark DNA? So even human beings may be having some dark DNAs. Scientists have mapped human genes, but there may be some DNAs, some genes which we have not been able to map properly. So once we develop technologies to better understand dark DNA or identify dark DNA, it would be beneficial for us. Like we know that we study evolution also through genetic analysis. Like whether we have evolved from some ape or not, uske liye genetic analysis is very important. If we have a lot of genetic similarities, then the evolution must have taken from them. We may have evolved from them or other living organisms also, a tiger, a lion, a species of tiger, a species of lion, where they have evolved from. Genetic similarity can play an important role in identifying this evolution. So right now we have not identified all the genes, dark DNA. So in future, when we identify all the genes, then we will better able to understand evolution. In future, when we identify all these dark DNAs, we will better able to understand the diseases which human beings are suffering from, because some of these dark DNAs may be facing some issues because of which we are having health concerns, health issues. This is all that we need to know about dark DNA. Understood? Clear? Okay. So we're done with DNA. Chromosomes, I was basically talking you about this. The chromosomes are these X-shaped.
The gene mapping is different for each and every organism.
Yes. So chromosomes I've already explained you. Chromosomes are the portion on which DNA is, like this was chromosome. If you cut open this, you will see that DNA is rolled over these things. These are chromosomes. And now come to RNA, ribonucleic acid. Do human beings have RNA? Human beings have RNA but not as genetic material. Our genetic material is DNA. Our genes are DNA, but RNA are there. Okay, so first of all, what is RNA? Ribonucleic acid. It's a chain of ribonucleotides where we'll have a nitrogen base, a ribose sugar, not deoxyribose sugar. Right? So it's a long chain of ribonucleotides where we'll have a nitrogen base, a ribose sugar and a phosphate group. Like DNA, RNA is also vital for living cells. So if you look at the shape and structure, so it doesn't have that double helix structure. Its shape is not that well defined. It can form double helix shape in some cases, but not always. So basically RNA exists in different shapes. Okay, now functions of RNA. What do RNA do? RNA क्या क्या करता है? So first of all, you know that RNA will be genetic material in some viruses. So in some living organisms, RNA is the genetic material. Fine. Second, the main job of RNA is to transfer, like, let me explain this to you in a simpler way. In the nucleus, we have our genetic material. Okay, let me draw a bigger cell. Let's say this is one single cell. In the nucleus, we have our gene, our DNA, our chromosome. Like in the nucleus, we have our chromosome. On chromosomes are DNA. On DNA is gene. Right? Where is protein manufactured? Protein is manufactured on the surface of endoplasmic reticulum, some other organelles, that is outside nucleus. Proteins are manufactured outside nucleus. We have to bring this information, what protein has to be manufactured, from inside the nucleus to outside the nucleus. Because it's inside the nucleus, we have the information, what protein has to be manufactured. But protein is being manufactured here. Ribosomes play a role in manufacturing of protein. Ribosomes are considered factories of protein. Ribosomes work on the surface of endoplasmic reticulum. So we have to bring this information to ribosomes, and this role is done by what is known as messenger RNA. So another function of RNA, what is another function of RNA? Transferring genetic information from nucleus to ribosomes, the factories of protein. So one function of RNA, in some living organisms, they are acting as the genetic material, they are storing the genetic material in viruses, in some RNA viruses. Here, they are acting as messenger, they are bringing information from nucleus to ribosomes. Come back, this is what we have discussed here. The main job of RNA is to transfer the genetic code needed for creation of proteins from the nucleus to the ribosomes. Ribosomes, which are factories of protein. This process ensures that DNA is always protected inside the nucleus and DNA doesn't get modified. Messenger RNA will bring it out. Then there are some RNAs which can also act as enzymes. These RNA enzymes are also known as ribozymes. So enzyme related properties are there. Okay, so in the manufacturing of protein, there are three types of RNAs which are used. Messenger RNA, messenger RNA तो आपको समझ में आ गया. It is bringing the information from genes from inside the nucleus to ribosomes. rRNA and tRNA. Let us also understand what this rRNA and tRNAs are. rRNA is nothing but ribosomal RNA. तो ribosomes जिसे हम बोल रहे हैं something that makes protein, factory of protein, they are made up of some proteins and ribosomal RNA. Those are RNAs. Then there is tRNA, transfer RNA. What does it do? For this, let us first understand how protein is manufactured. How is protein manufactured? So this is the nucleus here. This is the nucleus. This whole part is the cell. Inside the nucleus, we have genetic material, messenger RNA. Look at this. Messenger RNA is bringing the information out which protein has to be manufactured. These two subunits are subunits of ribosomes. Ribosomes which are factories of protein, which will manufacture the protein. These ribosomes get attached to the messenger RNA and read the information on messenger RNA. They read the information on messenger RNA. Everything is understood till now? Now, what do transfer RNA do? Transfer RNA, this is transfer. On transfer RNA are attached amino acids. They bring amino acids to messenger RNA. What is a protein? Protein is a chain of amino acids. One amino acid, second amino acid, third amino acid, fourth amino acid. In this way, protein is chain of amino acid. So how will you make protein? You will make protein by arranging amino acids. What protein to be formed is the information which is available to you in messenger RNA now. It has brought the information out. It took this to the factory, to the ribosome. Now transfer RNA, on it is attached amino acid, will bring this to ribosome. Ribosome will take the amino acid which is required and free the transfer RNA. This transfer RNA will get attached to another amino acid. In this way, ribosome will form this chain of amino acids and thus protein. So what was the purpose of transfer RNA? Transfer RNA transfers amino acids to ribosomes. Messenger RNA brings the information. rRNA is something ribosome is made up of, and tRNA, transfer RNA, is transferring amino acids to ribosome so that ribosome can form a chain of amino acid. That is the protein. How is protein being manufactured in your body? This is how protein is manufactured in your body. Role of RNA, messenger RNA, transfer RNA, rRNA, all those things are being done here, are being covered here. Understood? The details are there in your notes. For those of you who will be watching it on YouTube, please go through the description. In the description, we have the notes attached. Fine? Done? Okay. Now let's go back.
So when we talk of protein, is it the similar protein that we have in the food?
Yes.
Then the protein we get from the food, our body need not to manufacture that.
No. See, yes. We get amino acids. Using that, like, see, understand this. The protein that you get from chicken, does it form your black hairs? No, it has to be converted into a form. So you get amino acids, consume them. Using those, proteins get manufactured. Right? Like when you are eating flesh, chicken flesh, it doesn't directly convert it in your flesh. It is consumed by cell. And then using the nutrition from it, chain which is required in your body would be formed. How will that be formed? Based on the gene that is there, based on the genetic information that is there. Right? Understood? Okay. So mRNA, rRNA, tRNA, you have understood all these things? mRNA, rRNA, tRNA. Now let us understand one more thing. What is transcription? Formation of RNA from DNA. That is transcription. Simple. mRNA is bringing the information outside. Right? How is mRNA being formed? From DNA. DNA has the information, usko mRNA has to take out. So mRNA is formed based on that information. So RNA is formed from DNA by a process known as transcription, and here enzymes like RNA polymerase are used. There's another term that you should know, transcriptome. What is transcriptome? Now transcriptome is this. Let me again. In this cell, all the different mRNAs you have, they together are known as transcriptome. So understand this. Suppose you have gene 1, gene 2, gene 3, gene 4, gene 5, gene 6, gene to gene 20,000. You have 20,000 genes in your DNA. Let's say this is the situation. But if you do not have messenger RNA corresponding to that gene, will that gene will be able to express itself? No. You have gene for a particular protein 1, but for protein to be formed, a messenger RNA should be there which will bring the information to ribosomes. Otherwise, even if you have that gene, it will not be expressed. Expressing of gene means corresponding protein of that gene being formed. It will not be expressed. Coming back to this, so what is transcriptome? Transcriptome is set of all the mRNAs in a cell or all the mRNAs in that living being. This basically tells you which are all the genes which are going to get expressed, which are all the genes which are going to whose protein is going to be formed. Transcriptome. Fine, did you understand it, Kapish? Okay, look at the definition. Where is it? Transcriptome is the set of all messenger RNA molecule in one cell or a population of cells. In a population of cells also, like in one cell that mRNA may not be there, but if in another cell, if it's there, that protein can be formed by your body. Okay, because transcriptome includes all mRNA transcripts in the cell, the transcriptome reflects the gene that are being actively expressed at any given time. If mRNA is not there, that gene is not getting expressed. If mRNA is there, that gene is getting expressed. Fine? Loving it? Enjoying it? Understanding it? This was also asked in preliminary examination. What is transcriptome? And everyone was like, it's so simple. Now you know, it's set of all the messenger RNA. Because you know what messenger RNA does, you know. Transcriptome will basically tell which are all the genes which are getting expressed. It's a simple, very simple question, but people are generally not prepared for this. Now, let's attempt some more questions. We have looked at the process of manufacturing of protein as well. Okay, let's also look at RNA interference technology before we go to attempt some more questions. Now see, you know what messenger RNA is there? Now suppose there is one gene 1 in my body which is creating problems for me. There is one gene 1 in my body which is creating problems for me. If I interfere with the messenger RNA of that gene, will the problem being created by that gene stop? Because the protein associated with that gene will not be formed. Rna interference technology. You interfere with RNA. If you interfere with messenger RNA, you in a way are preventing the expression of a gene. So, RNA interference technology is a biological process in which RNA molecules inhibit gene expression or translation by neutralizing targeted mRNA molecules. mRNA molecules are neutralized so that genes will not be expressed. It is also known as co-suppression. Co-suppression. Post-transcriptional gene silencing. What was transcription? Conversion of formation of RNA from DNA. So RNA has been formed. Transcription ho gaya. After that, you are silencing it. Post-transcriptional gene silencing, PTGS. It's also known as quelling. So in 2000, I think 18 or 19, RNA interference technology was also asked in preliminary examination. Again, everyone was confused. But if you will study with fundamentals, it will be very, very simple. Okay, now what are the functions and applications of RNA interference technology? So one is, it's a vital part of the immune response to viruses and other foreign genetic material. What happens is, understand this. Now think of this aspect. COVID-19 disease. It's caused by which virus? It's an RNA virus. That virus takes help of certain protein on your cell to penetrate, right? If, I don't know whether this is being done or not, if we interfere with mRNA of that protein, can we prevent virus to be affecting you? So theoretically it's possible, practically it has not been done. That's why we do not have a foolproof solution yet. Okay, so RNA interference is a vital part of the immune response to viruses. Immune ka response to viruses and other foreign genetic material, especially in plants, where it may prevent the self-propagation of transposons. RNA interference has an important role in defending cell against parasitic nucleotide sequence like viruses. It can be useful to study the function of a gene. How can it study the function of a gene? Again, use common sense. mRNA, stop it. The protein formation, some protein formation would stop. You will understand, okay, this was the function of that gene. Interfering with mRNA will lead to that the protein whose formation has stopped is the gene, is associated with the gene whose mRNA has been interfered with. Okay, now let us, before we jump on to the next topic, let us attempt some questions. Okay. Consider the following statements about DNA profiling. First, DNA profiling is a forensic technique used to identify individuals by characteristics of their DNA. Second, DNA profiling determines gene functionality and position in the chromosome. Dusra, it was not DNA profiling, it was gene mapping, where gene was mapped to the protein, associated protein. It was not DNA profiling. Understood? Okay. So which of the above statement is our correct? The answer is one only. Fine. Come to question number 4. Consider the following statements about dark DNA. Let's look at the two statements and you tell me which are correct. The sequence of DNA which causes genetic disease are known as dark DNA. It may or may not cause disease, right? Dark DNA are some DNAs which are which we have not been able to identify exactly, but they're causing disease not necessarily. The one is wrong. 2. These sections of DNA have not been sequenced and they don't encode any protein. They encode protein. They have not been sequenced, they have not, but they encode protein, right? You people have understood things. I'm happy. You're enjoying this? You feel like this class is adding to your overall understanding of the subject? And we have not used any hardcore technical terms, technical things here. These are basic information which we study till class 10. Only using that we are studying everything. So what should be the answer here? Both are wrong. Answer should be D, none of the above. Fine? Okay. Come to question number 5. Consider the following statements about RNA. Okay, let Priyanka answer this. Priyanka ji? Can it? Where does it store genetic information? Some viruses. Nice. How? Nice. So RNA does play an important role in manufacturing of protein. Third? Yes, this is also true. See, DNA has to store like a lot of protein and all. RNA generally is storing information of virus. Viruses are small, basic. They do not form those many proteins. So RNAs are messenger RNAs carrying information of only one protein, one gene, right? So generally RNA have a much shorter chain of nucleotide than DNA. This is also true. So what should be the answer? D, 1, 2, 3. Come to question number 6. Which of the following pairs are correctly matched? Kashi. First one is correct. Kadhe. What was mRNA? Wait. What message is it carrying? It carries from inside the nucleus, which protein has to be formed? mRNA is carrying that information, no, from gene to ribosomes. Transfers amino acid to ribosomes? No. Who transfers amino acids to ribosome? Transfer RNA. Transfer RNA is bringing amino acid to ribosomes, no? Abhi padhaya tha yaar. tRNA was doing that. Messenger RNA is bringing something else. tRNA carries information from DNA to ribosomes for manufacture of protein? Wrong. Ribosomes, factory of protein? 3 is correct, right? So answer is 3. B, 3 only. Fine. Let's go to the next question. RNA interference technology has gained popularity in the last few years. Why? See, use the knowledge that you have. It is used in developing gene silencing therapies. Is it correct? तो देखो A, C, D में से कुछ बचता है, एक एलिमिनेट हो गया, अब तुक्का मार सकते हैं, right? It can be used to develop hormone replacement therapies. Now see, for replacement of hormones, you will have to activate some gene, or you will have to add some gene from outside. mRNA is stopping something from happening. It will not be able to replace some hormones. So 2 is definitely wrong. 2 is wrong. Answer C बच गया. Sorry. It can be used to develop hormone replacement. Achha, 3 is wrong. I'm sorry. 3 is wrong. What was correct? 1 is correct. 3 is wrong. 3 is wrong. ये दोनों गलत हो गए. अच्छा, we are left with ACD. Second, it can be used to developing therapies for the treatment of cancer. Now see, some gene may be responsible for cancer. some gene may be responsible for some protein which is multiplying a lot and is causing cancer. RNA interference technology can stop that gene from functioning. So yes, so 1 and 2 are correct, 3 is wrong. You are left with 1, 2 and 4. You do not need to know whether 4 is there or not. It can be used to produce crop plants that are resistant to virtual pathogens. Again, उस वाले जीन को साइलेंस कर देंगे जो पैथोजन अटैच हो रहा है. तो आंसर इज 1, 2 and 4. A difficult question, but if you understand the fundamentals, you'll be able to attempt. एकदम blank नहीं रहोगे. See, what I could have done, I could have discussed each of these options in your notes, but that's not the right strategy. Understand the fundamentals and you apply that in your paper. Okay, let's go to the next question. Recombinant DNA technology or genetic engineering allows genes to be transferred. किनके बीच में gene transfer allow करते हैं? Okay, Kapish will say. Across different species. Is gene transfer allowed across different species? Santosh sir and Golden Langur, we're different species, no? No, we're same species. Different, no? But gene transfer is allowed. Bacteria and plant, gene transfer is allowed, yes. From animals to plants? All living organisms which have DNA, उनके बीच में तो except some viruses, all living organisms have DNA. सोचना भी क्यों पड़ रहा है? I spent half an hour in explaining this only. Except some RNA viruses, from microorganisms to higher organisms, yes. Bacteria is a microorganism. We are higher organism. Plant is a higher organism. Can transfer take? Yes. Or this is also asked in Prelims 2013. Now you cannot cram. अच्छा इनके बीच में हो सकता है, इनके बीच में हो सकता है, इनके बीच में हो सकता है. You have to understand. DNA is the genetic material in all living organisms except some RNA viruses. And therefore this. Fine? Loving it? Enjoying it? Let's go to the next question. Advantages of traditional hybridization over genetic modification. Let's study these things. We haven't studied hybridization and all those things. Let us study these things before we attempt other questions. Okay, let's go to these topics. Just give me a second. When is your next class starting? Gene editing. No. 3:30. No? Or let's end this class here. Let's end it here. Tomorrow we'll start at 1:30. So 1:30 to 3:30 you'll have your first class. From 4 to later you'll have your second class. Okay. जब तक निखिल सर कल्चर पढ़ाना चाहेंगे. Let's stop it here today. These are fundamental things. Understand it. From tomorrow everything would be factual. This has happened, that has happened. Once you understand fundamentals, understanding those factual things would be very easy. Okay, I'll see you tomorrow.
Hit the bell icon to never miss an update from Level Up IAS. Make sure you like, share, comment, and subscribe to our YouTube channel.